Professor Zuzanna Drulis-Kawa is a full professor at the University of Warsaw, Faculty of Natural Science, Poland. In 2010, she became the head of Department of Pathology, of Pathogen Biology and Immunology, Institute of Genetics and Microbiology, University of Warsaw, Poland. And in 2016 to 2021, she was the head of the Institute of Genetics and Microbiology, Warsaw, Poland. She demonstrates expertise in clinical microbiology and diagnostics, alternative antimicrobials, phage biology, and phage-derived enzymes interacting with bacterial surface glycans and biofilm. Her research is focused mainly on the antibacterial activity of phages and phage-derived enzymes such as the polymerases and endolysins specific to escape bacteria, that is Klebsiella pneumoniae and Pseudomonas aeruginosa. Professor Zuzanna Drulis-Kawa is a co-author of above 90 publications in international journals. During the scientific career, Professor Zuzanna Drulis-Kawa received over 20 grants from national and international research funding organizations. She was the main organizer of EMBO workshop, Virus of Microbes 2018 conference held in Warsaw, Poland. She is the president-elect of the International Society for Viruses of Microbes for 2021 to 2022 year. Please join me in welcoming Professor Zuzanna Drulis-Kawa to give her talk. You're welcome, Professor.
Okay, shall I start now?
Okay, yes, Professor, you can start now.
Thanks a lot for the kind invitation and presentation of myself. As already mentioned, that I'm clinical microbiologist, and I used to work in a hospital in clinical lab. So that's why I was very worried about the emergence of drug-resistant bacteria. So then when I switched to the university, I started working on alternative therapies. So today I'd like to show you our recent achievement, let's say, published results in a model of Klebsiella pneumonia, because these bacteria start to be very severe and isolated from most of the clinical relevant samples. So let me start sharing my screen. Okay. All right, so tell me if you can see my presentation now.
Looks great.
Okay, perfect. So today I'd like to tell you about probably of the possibility of using phages and phage-derived enzymes against Klebsiella pneumonia. And before we start about phages, so let's say how severe Klebsiella pneumonia is, because this bacterium is producing a thick capsule, which can be composed of more than 100 of different types of polysaccharides. And this thick layer can protect bacteria against the immune system. So that's why we have so many problems, first of all, to develop an effective vaccine against this bacterium. This bacterium can propagate very efficiently in our organism, and additionally, they gain plasmids composed of multidrug resistant genes. So how the bacteria use the capsule? The capsule protects bacteria against physical conditions, dehydration. They help bacterium to adhere to different surfaces to form biofilm. But most important element is the capsule can protect bacterium against immune system. And specifically, it can inhibit complement cascade in our serum, that it can prevent opsonization of antibodies, and also can reduce the recognition by macrophages, so reduce the phagocytosis itself. So we may then think about or go to the nature to look for some solutions, how to fight this bacterium. So one of the solutions is a natural predator on bacteria, which is phages, right? And those phages have to also overcome those thick layers which bacteria is covered. So for that purpose, phages use specific enzymes, which are located directly on tail spikes or base plate or tail fibers. And those enzymes enable degradation of this thick layer. Phages can also have specific enzymes degrading LPS elements, as you can see here, shaving the O chain of LPS and getting finally the target, which is the final receptor before they can inject the DNA into the host. So when we look closer on those specific phages which infect Klebsiella especially, so we may easily recognize the phages producing such enzymes, which are located again on tail spikes or tail fibers usually. So we can see this is the bacterial lawn. This is the same plate, but incubated for 1 day, 2 days, or 3 days. And you can see that around the plaques, you can see some halo zone. Okay, and this halo zone is growing over time, as you can see here or here. And what does it mean? That enzyme, which is already dissolved in the agar, can diffuse and finally degrading the capsule on still living bacteria there. So what you see here, that these bacteria are taken from bacteria alone, unharmed, and those ones which are capsule-less are taken from the halo zone. So it means that the bacteria are still alive, but they do not have anymore the capsule around. So what's interesting, so when we look closer how the phages are organized in terms of the viral composition, so we may see that phages can be equipped in those specific enzymes as a one set of enzyme, meaning they can have one type of enzyme, which is specifically dedicated to degradation of specific capsule serotype. But phages may acquire a second set, like you can see here, the second set of enzymes, meaning this phage is able to attack two different serotypes. Okay, and what we found out that phages may acquire additional set, they can lose additional set by the let's say, adjusting to specific host. But some phages, as you can see on the right side, these are jumbo phages, which can even have up to 10 different types of enzymes, which are attached in a specific broom-like tail fibers. So it means that this phage is able to infect 10 different types of serotypes of capsule. So it has a broad activity against different Klebsiellas. So knowing that this specific composition or specific structure and acquisition of extra sets of those tail spikes or enzymes, we actually can use engineering genetic engineering to develop different GMM phages. So this is also our work when we using synthetic biology to exchange those specific fibers between different phages. So we took a scaffold of K11 phage, and then we removed or then we switched those depolymerases derived from other phages. So actually, we got GMM phages, which were able to target another type of phage of bacteria. So you can see this is the wild type of scaffold targeting K11 bacteria. But when we remove that and replaced by other depolymerases, it's turned out that this phage were able to infect different type of bacteria possessing K21 or K33 serotype of capsule. So now you see that we are able to modify phages and to mimic the evolution, you may say. So the first question what we asked ourselves was that can we use phage-derived depolymerases as antivirulent agents to synthesize bacteria to immune system? If so, we could help the immune system to fight the infection just to disarm bacteria from this very important shield which they use against immune system. So the assumption was very easy that if the capsule is protecting bacteria against the recognition of macrophages, so when we remove the capsule, the macrophages should start see the bacterium and should start to be able to engulf and to destroy destroy Klebsiella. So that was the one assumption. The second one was if we remove the capsule or we may shave the LPS, so we may provide the entrance or show the receptors for complement system activity, and then if we induce the system cascade, finally we got bacterial lysis. So then we verified this assumption using 3 different bacteria. We took Klebsiella K3 type, Klebsiella K21 type, 2 representatives, and we treat this bacteria with specific depolymerase. And what we found out that the assumption was true, but not for all strains. So it means if we took the depolymerase against K3, we finally got the reduction of number of cells. So the CFU was dropped down in the serum, but for that bacteria, which K21, the effect was not so significant, was not so impressive, let's say. So what we conclude later on after this experiment, that the efficacy of depolymerase treatment was serotype and strain dependent. So it means that not all depolymerases can give us such an effect. And why? Because in most of the cases, or in some cases as well, other elements like LPS could protect bacteria against serum activity. And what we also conclude from that experiment, that the capsule serotype was more important against the protection, meaning the composition of sugars was more important in a protection against complement-mediated killing than the amount of the CPS, meaning the thickness of the capsule. The second experiment, we verified the phagocytosis, the assumption that bacteria can be easily visible for macrophages. So what we saw that indeed, in some cases, the bacteria were easily recognized and engulfed, but not all of them. You can see one exception for this strain. Even if we remove the capsule from that strain, the macrophages were not increasing the engulfment. The second element we checked, the increased or the bacterial killing after phagocytosis. And in that case, we saw that, that indeed the decapsulation increased internal intracellular killing. And here I'd like to show you the results obtained by other group from Jim Bull team. They verified, they tried to find out if the depolymerases as a separate enzymes are effective in a treatment in a mouse model. And they compared the results versus phage application. So what they found out, that the treatment rate, the delay, let's say, was very important. So it means if we apply depolymerases, let's say at the beginning of the infection, the outcome is much better than when we are delayed with the application of enzyme. What next? That phages were superior to enzymes. It's quite obvious because the enzyme only disarm bacteria, but the phages disarm plus they kill bacteria, so they are more effective, and the immune system is, let's say, more supported by phages compared to depolymerases. And the route of administration, they consider also intramuscular versus intraperitoneal, and it was successful for some types of depolymerases, but not for this one. And the K1 capsule degrading enzymes provide highly successful when used even in leukopenic mice. So that's very important outcome. So our second and third question we ask ourselves, it was about the resistance emergence, because we usually consider the resistance as an adverse effect. In the case of antibiotics, if the resistance emerges, so we are very worried because we cannot any longer use that drug against the infection. But in a case of phages and phage-derived enzymes, we don't exactly know what kind of population will be selected after phage treatment of depolymerase treatment. If this is more virulent or less virulent, so we were checking that as well. So what we know about the resistance against phage infection? Here you have very nice and very important review wrote by Bernheim and Sorek group, and they provide a nice composition and comparison of different intracellular resistance against viral infection. We can have innate immunity, adaptive immunity, chemical defense, abortive infection, signaling system blocking, and many other not yet elucidated. But in fact, the first way, the first mechanism, the first strategy bacteria use against phage infection is actually related to the receptor. So it means bacteria modify, or let's say mask, the final receptor, protecting against adsorption of the phage. So you can see that in terms of protein receptor, and here you have protein receptor is hidden inside polysaccharides. So that's why phages develop specific way to overcome that barrier, like depolymerases. So if this is the capsule polysaccharides, so the phage is now equipped with enzyme degrading that polysaccharide. Okay. So thinking about this, we did a quite simple experiment. We treat bacterial population, Klebsiella, with depolymerase, and then we analyzed what kind of clones just emerge after the treatment. And you can see our recent paper that we analyze the modification in the genome composition. So what we found out that bacteria, the depolymerase selected for bacteria with the mutation in this specific gene. So this gene was actually knocked out because we saw the frame shift in the starting point. So as a result, bacteria stopped producing the capsule. So they were capsule-less. But important thing is that we did also the chemical analysis of extracellular composition of polysaccharides. And what we found out that the yield of CPS was dropped down, but then they start to produce more LPS. So meaning probably they replace the capsule with the long chain of LPS. So as you see, bacteria escaping the phage infection or depolymerase activity in a different way. But then we wanted to check if those mutants are less virulent or more virulent. So again, we checked the phagocytosis efficacy and intracellular killing. And so what we found out that bacteria which develop, they lost the capsule were more prone to phagocytosis, and they was combined with higher adherence and internalization to human lung epithelial cell. So you can see here the number of bacteria which were able to adhere to those. So you see how many bacteria, which this is the mutant, this is the wild type. So many more bacteria were able to adhere. Then we have the internalization after 2 hours was also increased for that mutant compared to the wild type, and as well and phagocytosis in the macrophages. In contrary, in the D panel, you see the survival rate in the serum, and we found out that there is no differences. So it means the bacteria were still resistant to complement activity. The next part, we wanted to find out if the phages can force bacteria to develop some resistant, and actually what kind of population is selected when we use phages. What the properties of mutants which survive phage infection. So in this experiment, we treat bacteria Klebsiella biofilm. These are 2 strains we treated with different phages, but both of them targeted the same serotype, K33, meaning they were specific against those 2 strains. So what we did, we treated, then we removed the resistant clones, and those clones were characterized phenotypically, and we did the whole genome sequencing, and finally we checked the bacterial growth suppression by phage cocktail. So what we found out, it's turned out that again, the accumulation of mutations in those clones which survived phage infection was found in the CPS cluster genes. So it means they were found in the genes encoding this specific protein, similarly as for depolymerase activity, and also in the gene encoding WZC gene, meaning the transportation of sugars outside the cell was stopped. So that's why we found that most of the bacteria were capsule-less, but we also found that some of them become mucoid. So as you see, the bacteria developed two ways to modify the receptor. In one way, they remove the capsule, and the second way they choose, it was overproduction of the mucoid polysaccharides, which differs in the composition. So actually bacteria again become resistant. And what we checked, we also were curious if the production of mucoid variants were the same in these two different strains. And it turned out that this is also strain dependent. More of the KP, this yellow strain, develop more mucoid versions versus the gray one. And what I think about, what I mean about hypermuco viscosity, let's move the movie. Just a moment. Okay, so you can see here how we remove the wild type strain. It's very easy to just remove from the plate. And this is the mutant, hypermucoviscous mutant. Can you see? It's attached very strongly to bacterial, to media surface and forms long stripes. So that's the test just to how we can find out that the strain is hyperviscous. Can you see a huge difference? Okay. And another interesting outcome we saw in terms of one of those strains, that half of the clones which develop resistance to phages become or switch from multidrug resistance to full sensitivity. You probably recognize this double disc test, how we detect ESBL enzymes production, meaning extended beta-lactamases production, specific formation of phantom zones. So this is the wild type, and the clones which were able to isolate were fully sensitive to the drugs. So when we checked also the MICs for different drugs, so you can see the wild type high level of MICs, and for the clones which were resistant, you can see a huge drop in MIC values, right? Can you see? So they become almost fully sensitive. So later on, we checked the composition of the sequence of resistant plasmids. So you see on the left side, this is the wild type, and these are the clones. And those green clones, you see these white gaps in the sequence. So this was actually the deletions, and those deletions contain most of the resistant genes encoded on that plasmid. So suddenly bacteria switch from being full resistant to become full sensitive version. And in the last step, we were also curious if applying the phage, which recognize the capsule, like here, those red ones, and getting resistant bacteria which are capsule-less, are they more or start to be sensitive to other phages, like those blue ones, recognizing alternative receptor? So it means, can we use more effectively the cocktail composed of two phages? One is recognizing the capsule, and the second recognizing alternative receptor, which is hidden beneath the capsule. And what we found out that when we applied the phages targeting capsule, so we were able to suppress the growth on some level. When we use those blue ones, those blue phages alone, let's say, those phages were not able to propagate on encapsulated bacteria. But when we combine this one with this one, as you see, we were able to reduce the growth very efficiently. So you can see this also in the A panel, that the blue phages alone were not able to stop bacterial propagation. Those pink ones, or the red ones, we see the suppression, and suddenly resistant clones start to propagate, right? But when we use the cocktail, so you can see that we efficiently inhibit the growth of bacteria. And we may also ask us, because the biofilm is one of important aspect as well, the same as in clinics in the common life. So can we use phages or enzymes against biofilm? So the question is yes, or I'm sorry, the answer is yes. And considering the very complex composition of the biofilm, here we have polysaccharides, extracellular DNA, outer membrane vesicles, bacteria at different propagation stage, and then we have phages as well there, and prophages released, and other elements, proteins covering the biofilm. So actually, yes, it's turned out that we may use phages because some of them can propagate in planktonic version, but also can propagate in those sessile versions. And against the elements of biofilm, we may apply specific enzymes. Against polysaccharide, we use depolymerases. Against proteins, we use proteases derived from phages. We also have LPS degrading enzymes against lipids or outer membrane vesicles. We may apply nucleases against eDNA and others. So to conclude, that the application of phages, or phage-derived enzymes, specifically depolymerases degrading capsule or LPS, we have to consider that the efficacy is serotype and strain dependent. But we confirmed that in some cases or some elements of the immune system can be enhanced by after depolymerases activity, or when we select resistant clones with enzymes or phages. And in vivo treatment was very effective, meaning also we may say that both phages and phage-derived enzymes are recognized as safe and effective. And what's important, that both phages and enzymes might be combined with antibiotics and effectively used against formation of biofilm or to eradicate the biofilm. So in general, and as we consider the phage application, we may say that we use lytic phages just to lyse the bacteria, but we may use the enzymes which derived from phages, one of them as depolymerases, as I said, degrading capsule or LPS, and later on we use them as antivirulent antimicrobials, you may say, sensitizing bacteria to immune system, or we may use lysins which degrade peptidoglycan, and the effect is cell lysis. So when we think about the current state the current stage of the development of alternative therapies. So you can see the report of World Health Organization that a part of it, part of the new developed drugs are actually related to non-traditional antimicrobials. And in that group, we have here phages and phage-derived enzymes. And some of them are dedicated against most threatful pathogens like Mycobacterium tuberculosis or Clostridium or Staphylococcus aureus. And an example of one drug was actually almost on the market is the extrabacase, which is the phage enzyme lysin, which caused the lysis of Staphylococcus aureus. So the clinical trials are almost done. And the next one, which is CF370, is dedicated to kill Pseudomonas aeruginosa. So as you see, now we enter a new area of non-traditional antimicrobials. And moreover, a new group just has been established in a frame of European Society of Clinical Microbiology and Infection Diseases, which gather people from phage ward and other non-traditional antibacterials. And if you want, you can join the group and be on time with news and what's going on there. And this group aims to foster, to establish a microbiology guidelines and to support phage application in clinics and out of the clinics as well. And I also would like to advertise our society, International Society for Viruses of Microorganisms. As you can see, how many people are already registered as members. A lot of people from America, North America, and South America, Europe as well. And then we cordially invite all of you just to join our society as well. So next, what our organization is organized, the different kinds of conferences, and the most famous one is Viruses of Microbes conference. And next year is going to be organized in Portugal, later on in Georgia, and in 2 years in Australia. So you are very welcome to join us. And I'd like also to acknowledge my collaborators from different centers, and thanks a lot for your attention, and I'm open for your questions. Thank you.
Thank you very much, Professor. That was a very, very insightful presentation. I personally have learned a lot because I had some interest in the use of phage-derived enzymes such as the polymerases, caffeine, biofilms, and this presentation gave a whole lot of clarity to my personal research on that. Right about now, we'll be taking questions. So we have a couple of questions in the chat box, and I will be reading them out for you. So the first one says, how could we identify toxic genes on the phage genome when we get isolate of a new type of phage? And then the second one says, if we do insertion of a certain gene into the phage genome, how could we check the stability of phage genome?
Just a moment, I have to read it. This is in the chat, right? How
yes,
we
could
identify toxic genes in the phage and when we get isolated new. For sure, we have to first sequence the phage. Otherwise, we are not able to identify those. But another problem is that most of the genes in the phage genomes are with unknown function. So could be the case that we will miss some information even if we do the sequence analysis. So, but without genome sequencing, I don't think we are able to detect toxic genes. I mean the toxic genes which could harm, you mean our host tissues, right? That's what you mean?
Professor?
Yes.
The second question says, in terms of designing a cocktail, would it be ideal if the burst size and latent time of phages are similar or close to one another so that competition among phages are lessened, or it is the other way around where the design should be a mixture of phages with varying burst size and latent time?
Just let me read carefully. In terms of design, it will be ideal if the burst size and the latent time are similar or close to one another so that the competition design should be mixed. Okay, so let's say it's difficult to say because as you saw in our experiment that in some case it's better to use cocktail sequentially or phage by phage sequentially. So it means they will not compete with each other for the host. So it means it's better to combine or to produce the cocktail of phages recognizing different receptors. You can then avoid the competition between phages. And moreover, bacteria will develop the resistance, like modifying the receptor, and suddenly two phages will be ineffective. So I don't think the burst size and the latent should be strictly the same. This is, again, very unique situation, and you have to consider this for each particular strain, I think. But for sure, we have to combine phages recognizing different receptors for sure.
Thank you very much, Professor. I have a question myself. I would like to know if we could use this phage-derived enzymes, such as the polymerase enzymes. I mean, without the virions, the phage virions themselves, just the biotechnologically derived enzymes alongside antibiotics to combat these biofilm infections.
Can we use, because I can't hear you very well, can we use the enzymes along with antibiotics to fight the biofilm, you asked?
Yes, Professor.
Yes, of course. I think if you use the polymerases, which are able to degrade exopolysaccharides, meaning just chopping the biofilm matrix, then you open the door, you may say, both for antibiotics, so they can reach bacteria hidden in the biofilm matrix. And moreover, when you remove that, you probably induced bacteria to switch to planktonic version just because they would like to escape the dangerous situation. If they want to escape, they will switch to planktonic ones or just to propagate intensively. And as we know, the drugs are very effective against propagating bacteria. So for sure, the application of enzymes degrading biofilm will enhance the activity of the drug, for sure.
Thank you very much, Professor. I think that's all for the questions that we have right now. Thank you very much for your presentation.
Okay, thanks a lot for invitation and for participation in that interesting meeting and event. Thanks.
Thank you very much, Professor. So
Thank you,
right about
Jessica.
now we'll be We'll be having a 1 hour break. We'll be having a 1 hour break to come back by
Okay,
3 p.m.
bye-bye.
Nigerian time.
Thanks a lot.
Bye, Professor.
Bye.